Thank you, Martha, and thank you for the introduction. I will share my screen. So thank you for the introduction again. It's always funny to have this introduction in the webinar organized by AXA because, of course, it's an audience that's a bit more Netherlands focused. So I did go to the Netherlands a couple of times in my career so far, in Utrecht and in Amsterdam, and it's just it's nice memories for me. So today I tried to make my talk a kind of complementary follow-up to the talk by Josep. What we heard about in the first presentation had a lot to do with the food we eat, how it's handled by the microbiome, and how it affects, in particular, the health of the gut. And what I will discuss today is more focused on what happens after that. So Josep already mentioned that those metabolites that come from our food or from the microbiome's activity are also absorbed by our system through the gut. And so that means they end up in our blood and they're distributed and they end up in all sorts of organs and having activities everywhere in our body. So how does that impact the host is what I will discuss in my presentation today. We will look very quickly at an overview of the use of metabolomics in microbiome research, and especially I will highlight certain interesting points on which matrix or sample type to use. Is it feces? Is it another type of intestinal sample? Is it blood, or is it even tissue samples? And what do you get out of those different matrices? Then we'll look at the links between the microbiome and disease and how to quantify the impact of the microbial metabolites on the host, and then look at a few examples of applications where metabolomics meets microbiome. So first, the place of metabolomics in microbiome research. So if you attend some of our webinars, you've probably seen that slide before where we see all the different ohms and the different omics that allow us to measure them kind of represented in relation to each other. And so we know that the microbiome is a huge contributor to the metabolome of the host. So even if you look at, for example, metabolites in the blood of an individual, you will find a large number of microbial metabolites, some metabolites that can come from the microbiome or from the host, because of course they share a lot of metabolites like amino acids and many, many more. And so when you measure the metabolome, you get a rich picture that you have to kind of decipher. And but the other omics, and this was also nicely represented in the previous talk, the other omics also have influences there and are interesting to study together with the metabolome. And of course, what's really interesting looking at the metabolome is also that you get a measure of outside influences. So besides the microbiome, whether it's gut or other microbiomes, you also will have either direct contributions of, let's say, the food itself or medications or other chemicals that we're exposed to, but also the impact of those different molecules on the metabolism that will influence the levels of metabolites. So it's why like all those arrows converge onto the metabolome, because a lot of things converge onto the levels that are quantified in whichever sample types that you choose to study. What's also important to remember is that the metabolome and also the gene products of the host also have an impact on the microbiome. So the same way that the microbiome influences us, we influence our microbiome. So lots of complications there, but what's important for me in that picture is that these things really communicate with each other and influence each other. And I'll get back to this in a couple of slides as well. So typically, if we think of combining metabolomics with microbiome research, you would have a typical metagenomic study where, and Josep introduced an example of this, where you get, you sequence the metagenome using one or another method, and then you get a taxonomic description of the population you're interested in, for example, of the gut bacteria that live in the intestine. And you also get a functional evaluation of what's happening based on, or what we think is happening based on the genes that are present in this microbial population. So it's one way to look at function of the microbiome, but of course it's a rather indirect way, because as we know, for the host or for animals, it's also true for microbiome. The presence of the gene doesn't necessarily need that the protein is expressed, and it doesn't necessarily mean that the activity at the metabolic level is happening in the way that we can expect just based on the gene levels. And so for that, it's really interesting to combine metagenomics with metabolomics, because with metabolomics, of course, you get directly the metabolite levels. So depending on which matrix you choose to measure your metabolomics, you can combine measurements in the exact same sample. For example, in feces, you measure your metagenome and your metabolome in feces, and then you have two sets of organisms and molecules that are present in the same place at the same time, and you can have a deeper interpretation of what that could mean. But I would also argue that measuring the metabolome in the blood can be a really interesting thing to combine with metagenomics, even if you measure the metagenomics in the feces. And I will get back to that in the next couple of slides. But in the blood, of course, you have all the metabolites that have been absorbed. So after they've had their effects on the intestinal cells and also on the immune cells around the intestine, they can still be absorbed. They're in the blood and they will be distributed to all the other organs. So changes in the population, let's say in the gut microbiome, can still have a quite large impact on the host, and that is somehow reflected by the levels of those metabolites in the blood. In both cases, you can detect rapid changes, and I think if you've ever either performed or read studies about, for example, diet intervention, you can see that after a few days or a few weeks, you can see really strong changes both in the microbiome and in the metabolome. So for that, it's also nice to combine those two omics together, because they have a similar kind of quick pace to measure changes in the system that is studied. And really the main added value of combining the two is that you get this direct measure of the function of the population that you're studying with the metagenomics. So you get much closer to quantifying the real impact on the host, and you combine not only the microbial metabolites, but also the changes in the host metabolites. So already how the health or at least the physiology of the individual can change. And of course, that's true both in humans and in animal models, and with very interesting applications in cell models as well. And one paper that's really interesting for this, especially for people who don't quite believe in this relevance of the blood metabolome in combination with the gut microbiome and with metagenomics, is a publication from the Institute of Systems Biology in Seattle, where they measured alpha diversity the usual way in feces, and then checked this against several standard clinical lab tests and against proteomics and metabolomics in the blood. And what they found is that 45% of the variance in this alpha diversity could be explained by a subset of 20 metabolites that were measured in the blood plasma. So that's a huge part of the variance of the alpha diversity. And what's also interesting is that out of those 40 metabolites, only 13 were actual microbial metabolites. So it's not just that you change the microbes and you're going to change which metabolites are present. The rest of the signature were metabolites from the host. So it's a combination of the actual microbial metabolites and the impact that they have on the host that allow to explain almost half of the variance in alpha diversity. Also, interestingly, the clinical tests and the over 200 plasma proteins that were measured in the same study did not manage to predict alpha diversity accurately. So this is also a case for the use of blood as a matrix in metabolomic studies, also in combination with metagenomics, and also for more technical or practical reasons. If you've ever worked with feces samples, you know that it's far from being a homogeneous sample type. It's complex to sample, you have to homogenize often, and it's difficult to have a standardized sampling method. And there are lots of people working on devices that help with this, but it's still difficult to have a workflow that allows to have a standardized way of sampling, so that when you go and quantify the metabolites, you have results that are comparable. And so for that, blood, of course, is much more practical. From the point of view of the sampling itself, I know, and probably you know as well, lots of people who would still rather go and have their blood drawn by their doctor than collect their own feces for an experiment. And so this aspect also has to be taken into account for certain experiments, and for that, blood can also be advantageous in a study design. But overall, I think for me, the main argument to study blood, and again, I have nothing against feces for this, but like really that people don't overlook blood in their study design is really that you get extremely interesting biological information when you combine blood metabolomics with feces metagenomics. And so to have a global overview of these different matrices and what we can get out of them, I have here, so from the feces, which is like, if you study gut microbiome, is the primary matrix that you would think about, although especially if you study gut microbiome, you may be at this stage more interested in collecting directly the inside of the intestine with different devices. And I think this is also really interesting if you can collect a part of the chyme and then analyze that with metagenomics and metabolomics together in the same sample or the same set of samples. This is a beautiful new avenue that I'm really curious to see the future studies that are going to happen on that level. But of course, feces is convenient to some extent and has some interesting information to it. But let's not overlook blood, which contains the microbial metabolites that are absorbed and signals on the impact that the microbial changes will have on the host. But also, there are lots of studies that look into tissue samples. So you can, of course, collect a biopsy, or if you're using animal studies, of course, collect an organ, and then analyze both the microbial and the host metabolites in that. And I will show you a couple of studies where, for example, there are microbial metabolites that are perfectly quantifiable in brain samples from humans. So nothing is stopping anyone from looking for microbial metabolites in organs. And really what matters is what you're studying and where you want to look at the impact of your experiment. So let's look at a couple of slides on the link between microbiome and disease before we look at more specific applications. I've already presented this in this Benelux webinar, but still I wanted to remind for the people who don't know about it that last year we released a white paper on the common origin of complex chronic disease. And so you can find this on the biocrates website very easily if you type chronic disease or white paper, actually. And what we discussed there is how diet and its effects on the microbiome associate with today's chronic diseases. And what we see is that what we did is we proposed models, so mechanistic models, for priming for complex chronic disease. And this included cancers, autoimmune disease, and neurological and psychological diseases. And these models serve as proof of concept for the application of metabolomics in different contexts. So we describe how you can use metabolomics, of course, to anchor the significance of the microbiome on a mechanistic level to different diseases. We also discuss its relevance to support the design of future studies, to find new druggable targets, and also to leverage intervention in that field, which have to do with diet and potentially supplementation, but also drug intervention, also so for nutritional studies, but also when one studies drug efficacy and patient response. So there are lots and lots of examples in that white paper. I encourage you to have a look if you haven't yet. And I will show you two examples here in this presentation. The first one has to do with the role of metabolism in the brain, but always starting with diet and microbiome as the source and the origin at the beginning. So if you look at the graphic on the right, you see that the left side is a bit darker, and this is where we describe the diet-derived and or microbiome-derived metabolites that we then link to the pathophysiology of the disease on the right-hand side. So you see that for this model on depression, for example, we looked into long-chain saturated fatty acids that you see in red on the bottom right. These get incorporated into triglycerides and acylcarnitines and all sorts of lipids that are going to increase inflammation. And we also looked into the effects of branched-chain amino acids like leucine or valine that are known to favor insulin resistance, and at bile acids, and especially the ratio of secondary to primary bile acids that is, of course, heavily influenced by the gut microbiome and which populations we have in there. And what was really interesting for us was to link that together with metabolomic studies of depression that showed in several independent studies that ADMA, methylated derivative of arginine, is increased in depression, in patients with depression. And so combining all the different paths linked to inflammation, linked to different types of signaling through different transcription factors and receptors, we suggested a model where we would link this increase in ADMA to a story that is currently not very clear in the field of depression, is whether nitric oxide synthesis is increased or decreased in depression. And putting the story together, we realized there could be a model where at certain times in the early stages of the disease, nitric oxide would be increased in part through an increase in inflammation. But over time, ADMA levels would accumulate, and this would lead to negative feedback that would decrease the production of nitric oxide. And so that could explain why there's a lot of literature with completely opposite findings on nitric oxide levels in depression. This is the first example I wanted to show today. And the second one, I will not go into as much depth, but I wanted to show here both because it's looking at different metabolites, and I think it's interesting, and actually it's a good link to the metabolites that Josep talked about in his talk previously, and also just to give a primer for the impact of these microbiome metabolites on the immune system, which is a very important aspect of how the microbiome influences host health. So if you look now at the dark box in this diagram, which is on the top left, you see that the metabolites that we look at here are, again, the bile acids, but also LPS, which is a typical part of certain bacteria that has been involved. So different types of LPS have been linked to the risk of developing type 1 diabetes. And we also linked in our model how the reduced levels of short-chain fatty acids, indole derivatives, and polyunsaturated fatty acids could all converge onto a modulation of the immune system. And what we show in this diagram is that we hypothesize a mechanism where the immune system is first inactivated, and this gives the possibility for certain mistakes in immune training to occur before the immune system is completely overactivated and then propagates the mistakes that have been that have occurred in the meantime. So we link then this model to 3 autoimmune diseases, IBD, multiple sclerosis, and type 1 diabetes. So if you want more details about this, please take a look at the white paper on our website, and feel free to contact me if you have any questions once you've read it, because I'm always happy to discuss the science with people. But let's look at how we can quantify this impact. And of course, if the impact is seen in the metabolome, then quantifying the impact means using quantitative metabolomics. So this is what we really try and support our customers to do at biocrates, using our standardized kits that are quantitative. For the one that I show here is the MXP Quant 500 XL. It allows to quantify up to over 1,000 metabolites, and that's a mix of small molecules and lipids. And I will talk about this in the next couple of slides, and Bijan mentioned it. We also have a tool that calculates sums and ratios of metabolites, and so with that you get to almost 1,500 markers that can be quantified. If the metabolites are in the sample and can be quantified, then you can have up to 1,500 markers. And so you see, you can link the metabolites that you measure with the kit. You can group by functions, you can group by certain aspects of the pathophysiology, like inflammation or insulin resistance that I mentioned in the previous slides. And you can link that to different organs and different organ-related diseases. And in the end, what you do is that you describe biology, and you can describe things like aging, or you can describe a specific pathology, starting from the very small and then going like spreading towards the more complex implications of these changes in the metabolome. And MetaboIndicator, so is this tool that for each kit that we sell automatically calculates sums and ratios of metabolites that give additional data points, but they're not just random ratios. For example, we don't calculate all the possible ratios. We calculate the ratios and the sums that have previously been used in the literature and in the field, and because they were shown to bring some insights. So, for example, here we have 4 sums and ratios that could be related to some extent to the activity of the gut microbiome. We recognize on the bottom right the secondary to primary bile acid ratio I was talking about previously. Also, the kynurenine to tryptophan ratio is a very well-known one to assess inflammation a bit more finely. And there are all sorts of such ratios in this tool. And what's really interesting when you get to the point of interpreting your results is that each one of these ratios has one or more literature references assigned to it. So you can immediately go in the literature and figure out how this has been used by other people. And today we also have a way to categorize those metaboindicators. For example, if you're interested in the microbiome, then you can go and just click the microbiome category, and then you see all the ones that could be calculated in your experiment and that relate to the microbiome to some extent. Here you have a list of some of those sums and ratios linked to the gut microbiome. Of course, a lot linked to bile acids, because there are 14 bile acids in the kit that I mentioned, but there are others linked, for example, to TMAO or to P-cresol or to indoles that give you additional insights in addition to the quantification of the single metabolites. And so to finish, I wanted to look into a few applications where metabolomics meets microbiome from the literature and from the work of some of our partners and customers. So here I start with an overview looking at this impact on different organs, because depending on the research that you do, you might be more interested in one organ, and sometimes it's a little bit boring to hear just about the brain when you work on the lung. But actually, whatever you do, I think you can always find to some extent a link to the microbiome and how the metabolites generated by the microbiome can impact the health of the system that you're studying. So I already mentioned it, but the immune system is really one, if you call it an organ, and I would call it an organ, is really an organ that, or a system that is deeply influenced by the gut microbiome through the metabolites that they generate, and actually not just the gut, the microbiome. For example, tryptophan derivatives, short-chain fatty acids, we've mentioned them already, are essential in the training of the immune system. Indole derivatives, for example, also associate with pain and certain inflammatory diseases. If you're more interested in the heart, you may or may not know that indoxyl sulfate, TMAO, and other metabolites that come from the microbiome, at least originally, linked to, for example, kidney damage in mouse models of hypertension. If you're interested more in the gut, looking into the fecal levels of bile acids can be interesting. If you, for example, work on Crohn's disease, where it's been shown to change in pediatric patients and to associate with the microbial community structures. The example of the brain that I mentioned before is from this publication by Kaleski et al., where P-cresol sulfate and other microbiota-derived metabolites were quantified in the brain of patients with Parkinson's disease. If this is a topic you're interested in, I encourage you to listen to the Metabolomics episode where I interview Karel Kaleski, the first author of this publication, You can find this podcast on the internet or on the biocrates website. It's called The Metabolomist. And so it's a podcast where I interview scientists about their work with metabolomics. And so this specific paper by Keleski et al. Is discussed there. There are also many studies showing that short-chain fatty acids protect the brain by strengthening the blood-brain barrier. The lung is also an organ that is influenced by what we eat. And so if this is something that interests you, this is one paper that you can look into, linking short-chain fatty acids and p-cresol sulfate to pulmonary vascular remodeling. And then the last example I want to give here is linked to the pancreas, and more specifically to pancreatic cancer. This is a study that I will describe in the next slide, where a metabolite of tryptophan could modulate the response to chemotherapy of an animal model with pancreatic cancer. So let's look at this study, and I do show this slide from time to time because I think it's really a beautiful example of how one can use metabolomics as an inquisitive tool to try and find the most interesting pathways or molecules to look into and to design follow-up experiments. So in this study, by German researchers, the question was, can we improve the responsiveness of pancreatic cancer patients to treatment? So they had a small cohort of a few dozen participants, and less than half of them responded to the polychemotherapy treatment that was given to them. And when screening the blood of those patients with the Quant 500 kit, what these researchers discovered is that 3-IAA, so indole acetic acid, was higher in the responders than in the non-responders before treatment. And so this pushed them to develop a mouse experiment where they transferred the fecal microbiota of the responders and non-responders into a mouse model, gave the mouse model chemotherapy, and then checked the response. So the mice that got the responders' microbiome responded to the chemotherapy, and they could even increase this response by giving a high tryptophan diet to the mice. But the mice that got the non-responders' microbiome did not respond to treatment, similar to the human situation. The tryptophan diet did not help them, but what did help them was to supplement them directly with the microbial metabolite. There is some logic to this. If you don't have the bacteria to transform the tryptophan into 3-IAA, it makes sense that tryptophan doesn't help you. And so now the next step is to confirm this in a clinical trial in humans. We will see how that turns out. But so what we see in this example is that from the starting point in the middle, where you have about 50-50 responder, non-responders, depending on the supplementation that you give, you can go to the bottom part here where you actually turn responders into non-responders. And this I didn't show, but if you give tryptophan too long, you actually cancel the response of the responders. This was a really interesting aspect. So you have to, of course, be careful how you manipulate the response of the patients. But if you provide the right supplementation, you have the potential to boost the response of the responder group and also turn non-responders into responders. Am I short on time, Bijan?
Honestly, yes, a bit, but please.
Okay.
It's okay. It's okay.
Okay, I will finish with this slide.
We'll have some final conclusion.
Sorry, I get carried away. Thank you for keeping me in line. Okay, so I will finish with this one just to show that we can go towards 4P medicine, so really the medicine of the future, and improving therapies and also making them more personalized and predictive in many different ways, using what we know about the microbiome and the interaction it has with the metabolome. So the first example in red I've just discussed, so this can give us answers for personalized medicine and for stratification of patients for treatment options. FMT, so this transfer of the fecal microbiota, you can also predict who will be a good responder to that, for example, using metabolomics. In terms of participatory medicine, Prebiotics can be a very good way to help people take their health in their own hands in a way. So prebiotics are, of course, the food we feed our microbiome. So this is a very interesting avenue when you combine metabolomics and microbiome. And the last example, which will probably give us a discussion point with Josep, is postbiotics. So there was a study a couple of years ago where multiple sclerosis patients were given a supplementation with propionic acid. And this really helped reduce inflammation and the flares in this multiple sclerosis group. And so this would bring me to the question, can we really improve our health by chewing our food more? Because this was the one that increased the propionic acid synthesis. I will skip this one and get directly to the summary. So I think we've seen that metabolomics is a great way to transform the raw metagenomic data into actionable health insights. Feces can be a very interesting metric, so is tissue in certain contexts, but blood is really one that people should not overlook when they plan a microbiome study. And you get information both on the effect of the foods, of the diet, of the microbiome, and of the host. And there are standardized quantitative tools that are there to help us make the most efficient use of metabolomics in certain contexts. And I've shown you that there are many different applications to disease conditions that can be really interesting. Sorry for being a bit over time, and thank you for your attention.